Hi, everyone. Thank you for joining us. On behalf of Metabolon, I would like to welcome you to Answering Common Questions about Metabolomics. My name is Carolyn, and I'm the host and moderator for today's event. Before I introduce you to our presenters, I'd like to provide some brief instructions. The presentation has been muted, but we welcome questions that can be answered during the Q&A session at the end. To submit a question, please open the Q&A window at the bottom of your screen. We also will feature a poll at the end of this event and welcome your feedback. Now I'd like to introduce you to our presenters for today. They are Carrie Wong, PhD, Associate Director of the Discovery and Translational Sciences Group, and Michael Finelli, Business Development Executive. Welcome. The purpose of this webinar is to provide a brief introduction into metabolomics and address important considerations for a successful study. We'll provide this content in a conversational format. Our discussion should be about 20 to 25 minutes with a 5 to 10 minute time frame for the questions at the end. Carrie, the presenter role is yours.
Thank you, Carolyn. Just to introduce myself, like Carolyn said, my name is Carrie Wong. I've been at Metabolon for over 4 years now, and I work to help clients understand what the technology is, and on the back end, help them understand and provide meaning to how to interpret the metabolic data. Michael?
Yeah, so in business development, my primary role at Metabolon is to facilitate mutually beneficial research and clinical partnerships. Before we get into the common metabolomics questions, I would like to provide a brief summary on Metabolon. So we are a metabolomics contract research organization headquartered in Research Triangle Park, North Carolina. We have a global presence with respect to both our client base and analytical testing sites. Over the last 20 years, we've pioneered the field of metabolomics, establishing ourselves as a clear leader. At this point, we have scaled to over 200 employees, including analytical chemists, data scientists, and biochemists. Now I'd like to set forth our agenda. So today, Carrie and I will address some key questions about metabolomics in general, successful study design, how metabolomics fits with other data streams, and what the results of a metabolomics experiment looks like. Carrie, we often work with scientists who are more familiar with other omics like genomics or proteomics. For those individuals, could you describe metabolomics at a high level?
Sure, and this is something especially with metabolomics being one of the newer technologies. So metabolomics is the study of small molecules in any living system, biological system. And we really advocate for the studying of these metabolites because they lie at the intersection of internal and external cues. So what I mean by that is your internal systems, such as your genetics, proteins in the body, all will influence what the metabolite signature looks like. In addition to that, we will also be including in that metabolic signature inputs, external inputs such as environment, lifestyle, microbiome, diet, all of these different things come, and that's where the metabolites are able to integrate these multiple inputs, providing a very close picture of the phenotype. And really what I'd like to kind of expound upon is that despite being around for more than 20 years, metabolomics is only now becoming well known in the market. And so what we're trying to do here at Metabolon is really help people understand what the meaning of a metabolomic data set is, and help them from the beginning, including study design, all the way through interpretation to really gain the most out of their data sets.
Thanks, Carrie. Could you expound a little bit more on how Metabolon's approach to metabolomics differs from others?
Yeah, so there's generally metabolomics comes in two flavors. There's a targeted and a global approach, and we actually do both. So as the name suggests, a targeted approach means you know exactly what you want to look for, and you're not going to look beyond that 5 to 10 metabolite panel. When we take the global approach, we're going to cast a much wider net And this allows us to capture as much of the biological picture as possible. And oftentimes, this global approach can inform the targeted approach. So, for example, if you're looking for a biomarker, you might say, "Oh, I don't know what the best biomarker might be for a disease." And you start with a global approach. You see which metabolites differentiate between healthy and diseased states. And then you can use that information to decide, here's the best targeted assay that can be performed on a routine basis moving forward. And so oftentimes we try to encourage clients to start out with our global approach and then work with our targeted assay team to customize a targeted assay as needed that can be routinely run. And so when we take this global approach, which is really the bread and butter of Metabolon, we'd like to take two things into consideration. A, broad coverage, and B, data reduction. So how do we make sure that we have broad coverage of metabolites across metabolic classes? And we do this by, over the last many years, generating a library of authenticated standards. And so at this point, we have over 5,000 authenticated standards that we use at every time we run a study. We're screening against those standards in order to provide as much information as possible. And this library also allows us to reduce the data, and so we can take a chemocentric approach. And this is really important to confidently report on metabolites rather than ion features. So within a given study, you'll run a sample and there'll be thousands and thousands of ion features. And it's important to understand that a single metabolite has multiple ion features and vice versa. And so by taking this chemocentric approach, we're able to understand exactly what the compounds that we see are based on the retention time, mass, and spectral information, so that instead of giving you a bunch of information that you have to sift through to remove redundancies, we've removed those redundancies to give you just the metabolites along with their abundances.
Nice. As often the first point of contact at Metabolon with potential clients, there's one question that comes up an awful lot, and it's essentially, do you see compound X?
Yeah, that is one of the most, I have to agree with you, that's one of the most common questions we receive. And the way we do this is we do handle them on a case-by-case basis, and we can always check against what we have in the library. And what I often like to point out to clients under these circumstances is the value of the big picture. So yes, not only will we likely be able to detect the compound you're looking for, but if not, we often are detecting metabolites very closely related to compound X in this case. And this does a variety of things. A, it gives you confidence in that a certain pathway or metabolic node is in fact being impacted if you do detect changes within a pathway. Additionally, there's certain cases where some metabolites that may be the first thing that come to mind are extremely important for the health of an individual. And if you only are looking at that single metabolite, you may not see or detect differences that could be indicative of a subclinical phenotype. And so it's very important that we try and take a holistic approach until we have a little bit more confidence that a more targeted methods might be appropriate. And yeah.
That's well put. Kind of along a similar vein, a lot of potential clients are often interested in their specific disease state. They essentially ask, hey, I study this pathology. Will your platform be able to give us the answers that we're interested in? How would you address that question?
Yeah, so Metabolon has worked with over 500 different diseases and conditions, so it's likely we do have some experience in whatever you're studying. And that said, metabolomics can be used to study any biological system. And so even if we haven't worked with your specific disease, it's more important that we can understand exactly what question you're asking. I like to say that in any given area of research, you guys are the experts. You guys know more about that. But what Metabolon can provide is our expertise in metabolism. And that is something that together, our combined forces might be able to move your research projects forward. So what I like to start out with clients is understanding what their question is. Are you looking for mechanism of action? Are you looking for efficacy? Are you interested in biomarker discovery and confirming target engagement? Once we have those and we can really specifically and clearly articulate your goals, then we can sit down and Metabolon can help you design a good study.
Now, one of my favorite discussions that we have with clients is when we start getting into experimental and study design. Could you talk a little bit about, at a high level, what a good study design looks like?
Yeah, this is another very common question, and this is where I think it is extremely important to try and have these conversations up front. What's important when we're designing a study is to have good power and a well-controlled study. And so why do we want a well-controlled study? We want a well-controlled study because global metabolomics is dependent on relative quantitation. So the data is reported back as full changes from some sort of a control group. And that can be a baseline time, it can be a placebo-treated group. Again, these are all discussions you'd have with the study director to hash the specifics out. And then after we understand, then we know we have a good control group, we're going to move on and we want to make sure that the study is properly powered. And we've done power calculations with different research models to determine what numbers are optimal, rigorous, and acceptable. And that's what you see here. As you can imagine, as you change your research model, the variability also changes with it. So cell culture, a cell culture where there's not a lot of variability, you seed your cells, it's the same cells over and over again, we can get away with a less biological replicates per group. Then as we move into small animals, this includes mice and rats and such, we look to get groups greater than 10, but anywhere from 6 to 10 is acceptable and can provide excellent information. And then when we get into human studies, especially free-ranging humans, I know everybody on this call is eating something different for breakfast, and they're gonna have something different for lunch. Exercise, doesn't exercise, drinks, doesn't drink. All these are going to influence the metabolome. And so for the human studies, we really recommend powering them, above 50 to really get some very statistically powerful data. If you go below this, again, we want to talk about, make sure we're talking to our study directors about this, but there are different approaches you can take. If you're just interested in kind of looking for trends, then you go a little bit lower. You just know that you're going to have to follow up and on, as you would with any study, to find changes that might be biologically meaningful. I've also put together here a couple of examples of well-powered, well-designed studies for the different models that you might be studying. Up here we have a case cell-based study where you might be looking at a drug treatment, and you'll have 2 different doses of said drug as well as multiple time points. And this will give us an idea not just of what's happening to cells upon treatment, but also if there's an impact of time. And it's very important that we have the vehicle control, as I mentioned, as well as the 2 different doses. And here they had 5 biological replicates in the study. For small animal studies, such as mice, as you can see here, you might be, oftentimes these are 2x2 studies where you might have a wild type and some sort of genetic knockout or treatment, and then this might be combined with 2 different diets. And again, well-powered with 8. And then as we move on to the human studies, this is a study that we've done where they had a case and control. This is also 2x2 where they also divided by gender. And this is powered at 30, so it's not the optimal, but it is rigorously powered, and we can expect to see differences in what we might see with this group.
Under what circumstances could we get away with a lower powered study?
Oftentimes if sample types might be rare diseases, for example, and we will work with you to run those. And again, I cannot stress the importance of discussing this with your study director as soon as possible in the discussion. But we can work with them, and in those cases, we might say, all right, are there clinical manifestations that might or other pieces of metadata that suggest that there might be vast differences between these two groups, then a lower power might be more appropriate. On the other hand, if it is a very subtle phenotype where you're not expecting huge differences between the groups, that's when we really got to recommend either we power it up, maybe we add, we do take a paired approach where you have multiple time points from a single donor subject. And this will allow us to have a little bit more confidence in the data we do see, and set the expectation that, all right, maybe we won't reach p-values of 0.05, but we will see trends in the data that might give us important clues when combined with other data streams that something biologically important is happening, and we can follow through with those observations.
Well said. What type of sample types do we work with?
Well, there's not much we don't work with. We handle plasma. That's probably EDTA plasma is probably the most common material, but we take saliva, urine, feces. Additionally, we can extract metabolites from tissues that can often be important for understanding exactly what's happening at the site of infection or the site of action. We'll do flies, fruit flies, things like that, as well as plant. There's really nothing we won't at least consider.
Is there anything that clients need to do prior to submitting samples?
No, they don't need to do much. In fact, as a general rule of thumb, we really encourage people to kind of freeze their samples as soon as possible. So get them on ice, dry ice, ethanol, or liquid nitrogen, and then store at -80. One of the most important things is that these clients treat every sample, treat the collection method as consistent as possible. And what I'm going to show here is what Metabolon has recently generated are these steady success sample handling kits. And we're able to provide clients with barcoded tubes that they can put their samples in. And this is really nice because it helps us make traceability and automation a lot easier. And also on this slide, we have some sample quantities for recommended optimal results. You can see for biofluids, this would be your plasma, serum, urine, CSF. We request between 150 and 300 microliters. For cells, we request 100 microliters of packed cell pellet. And the reason we go with a cell pellet instead of cell count is because different cells are different sizes. So 10 million cells for one type of cell, 10 million cells for one type of cell isn't the same amount of biological material for 10 million cells of another cell type. And so we do know that if you scrape your cells, spin them down into those microcentrifuge tubes, and when you spin it down, that pellet gets that 100 microliter hash mark on that tube. That's going to be enough biological material for us to extract the metabolites and get optimal coverage. And then, as I mentioned, we also accept tissue, and the best practices recommend that we have 50 milligrams of tissue to work with on a per sample basis.
Perfect. However, what if a client can't meet the sample requirements?
Again, that's something that is often the case. We have tumor samples, things along those lines. Mouse tissues often are much smaller than 50 milligrams, and we can still work with them. I'm going to say this over and over again, but the key is to talk with us as soon as possible. The worst thing that can happen is say, okay, no problem, and then send us less volume than requested and just hope for the best, because then there's a lot of shuffling that has to happen, and we want to make sure we can give you the best data possible. So if we know about your challenges early on, we can work to overcome them and to make sure that we're focused enough to get the best data possible. So of course there might be some trade-offs. There might be slightly a decrease in coverage or increase in variability, but if we can talk up front, we can know what's most critical for the client, and so then the approaches we develop are going to protect your primary needs, and we can still move forward and, like I said, give you a lot of good data.
Often when we engage with new clients, they've already looked at their biological area of interest for some time, and they've used other omics methods. How do we combine metabolomics data with other data streams, essentially the proteomics, genomics, transcriptomics areas?
Yeah, now more than ever, I think it's extremely important that people understand the different data streams and look for ways to integrate them. Metabolomics is by no means meant to compete with these other data streams. Rather, it's meant to be complementary to other omics data streams, such as proteomics and genomics. It is just another piece of the biological puzzle that everybody's trying to put together. And so metabolomics can come into play and be very important and informative, because when it's combined with things like genomics, it's going to give you a functional readout. And this can often provide an extra level of biological insights. A lot of genetic variants don't have an obvious function or biological impact. So and when you do a genetic study, excuse me, you might see a lot of changes, and you don't know what to go after. It's just too much information. And so metabolites can often, changes in the metabolic signature can often be used to say, okay, here's some genetic insights that might be a little bit more useful. Here's where we should tackle next in our research. And these might be functionally important genetic variances. And remember that the metabolites are going to integrate both these internal and external cues, so you're going to get an even bigger global readout of the biological system as a whole.
Could you go into a little bit greater detail in terms of helping people understand how this looks in a multi-omics approach with regards to what a global metabolomics data set looks like?
Sure, I'm going to pull up the slides again. We do want to help. One of the goals of Metabolon is really to enable the clients to be able to not only understand their data, but interact with their data and use this data in a meaningful way to drive their research forward. And to this end, we've developed an online portal called My Metabolon that's going to give you various opportunities to interact with the data. We have a fireworks plot, which you see here. And this is a way to globally look at the metabolic signature as the metabolites are grouped by a super pathway. You'll see different colors here that's going to indicate if things are statistically increased or decreased relative to a control group. And then you'll see the size of the circles are going to reflect the degree fold change between two groups. And so this is a nice way, once stats have been produced, that clients can go interact with their data. In addition to kind of looking at on a super pathway level, the My Metabolon portal also provides the opportunity to examine the metabolites within their pathways. And so this is a pathway map of all the biochemicals, and you'd be able to zoom in. There's actually a video on Metabolon's website that takes you through the different capabilities of My Metabolon, so that you can become more familiar with how you can interact with the data that your study has generated. In addition to that, we don't want to just lob a bunch of data over the fence and then run away. We really want to help you and walk you through what your data means. And to that end, we have a group of PhD biochemists that provide interpretive reports. And these reports come in two flavors. We have a high-level report that might be just a quick study director taking a quick look at the data and trying to point you in the right direction of things you might want to follow up on, observations you might want to follow up on. In addition to that, we also have a deep interpretation. And this is where really your study director is going to hold your hand through the data analysis process. So we do the stats, and then we'll look through all your data and really investigate what these changes mean. We'll have conversations with you to understand your goals and the biological questions you want to answer. And then we're going to walk you through, we're going to set up a PowerPoint discussion to really discuss what we mean and what we think is happening. And it's an opportunity to be give and take process where you can let us know about things that observations that you see and questions that you have. And this is a really nice product to make sure that you are comfortable with the information and can integrate it with any other pieces of data that you might also be studying. And so that's the last thing I wanted to mention, that in the My Metabolon, it also provides the opportunity to kind of look at pathway enrichment scores. So you're not looking at metabolites as individual analytes, but also as a pathway to understand, is a pathway shifting in response to some treatment or genetic impact. I
really love this biochemical map. And that's probably because I'm less of a biochemist and more of a biologist. In this human plasma study, about how many compounds were detected?
Yeah, in this, good question. In human plasma, we're probably around 900 to 1,000 metabolites, both named and unnamed. So that is, as you can imagine, a huge amount of coverage. And I'd also like to add that while it's 900 to 1,000 metabolites, 8 different metabolic super pathways are covered in that. And so it's not just, we're not giving you a list of just one class of metabolites. We're going to cover multiple metabolic nodes. It's going to give you a very nice global picture of what's happening inside these cells or organisms.
Perfect. Well, those are all the basic questions I have, Carrie. I always love working with you, and your passion for metabolomics certainly shines through. Carolyn, if you have some questions, I think we're ready to address those.
Thank you both, Michael and Carrie, very much. I'm going to launch a poll really quick if you guys won't mind just looking at it and addressing and giving some feedback on it while we look over some of the questions. And again, if you have questions to answer, the Q&A button is at the bottom of your screen. So getting started with those. What is the most challenging aspect of metabolomic studies, both in terms of scientific challenges and from an operational standpoint?
I think, from an operational standpoint, one of the things that I cannot stress enough is to try and collect the samples in as a consistent manner as possible. We don't want people to, at one site, to collect in a fasted condition, and then at a second site collect samples in a fed condition, because there's going to that condition is going to grossly impact the metabolites. So it's very important that we try to be consistent as possible to remove variability. That's one of the operational things. Second operational aspect, or kind of overall in studying metabolites, is we're still waiting for the technology to catch up to be able to get coverage with low variability on a smaller volume. And that's because, unlike DNA or RNA, which can be amplified, you can't do that with metabolites. They are what they are, and we're measuring them in their native state. And so that's often why there's so far, and I know people are working on it, there's not a single cell metabolite assay that's going to give you 1,000 metabolites.
One thing I would add to that from an operational standpoint that we find really helpful is something we call a study success kit, where we essentially send you barcoded tubes, and you use that essentially to mark out your samples and different condition states. And so there isn't any confusion, and we don't end up with a sample manifest and a box of written on tubes and have to ask, hey, is this a P or a D?
That's great. Thank you. And speaking of sample collection, do preservatives or reagents in the transport medium impact metabolomics?
Likely, yes. A lot of, I know that there's a lot of tubes out there or collection methods that have DNA and RNA preservatives. And that is a question that we get often. And the answer is that those preservatives can definitely interfere with the data readouts. The reason for this is that although the RNA and DNA preservatives are RNA or DNA free, they are not protein free or salt free. And, you know, proteins have amino acids, and amino acids are small molecules. So there's always going to be those types of ingredients that can really impact your data, and that it makes it not interpretable. And so we have to avoid including things like those preservatives in the collection tubes.
Is there a way to tell which metabolites were produced by microorganisms in the body versus humans?
The million dollar question.
Yeah.
Metabolon has done quite a bit of research to determine which metabolites have been linked to microbial metabolism. Oftentimes there is a lot of overlap, but there are some metabolites, many of which are captured on our global platform, that are derived from the microbial metabolism. So if this is something of interest to you, what you would do is, as you're talking to your study director, before you send the samples in, you say, hey, I'm really interested in the microbiome. And we would make sure that in those interpretive reports that I was referring to, that we would include a section that says, all right, here's an aggregation of the metabolites detected in your sample that link back to microbial metabolism. Here depending upon the level of interpretation, we might include some facts about if they're involved in any disease processes or involved in the health of the organism in general.
In terms of patients in the ICU, there's no ideal way to control specimen collection. How do you suggest controlling for variability of clinical samples?
So, yeah, that's a tough one. We do ask that, you know, if the samples are treated similarly. I know that's hard. If we can track how long it took from, if there is a way to track how long it took from collection to freezing, then that's something that can be statistically addressed later downstream once the samples have been run. And then like I said, the best thing there is to, if possible, power that study. I know that's not always the ideal situation, but that's one to try and collect as many as possible. Another potential method for addressing that is if ICU patients, if it's possible to have a serial collection. So if can be 2 or 3 time points from the same patient, then you can start to look at within patient changes. So if you know that they were always taken by the same person or same hospital from the same patient, that can help weed out some of the variability that's going to be expected.
Yeah, the other thing I had, if you're working with multiple collection sites, as you send us those samples, please identify which sites they came from, and we could take an analysis that's separate from the biological question you're interested in, and see if there are changes in the metabolic profile, for example, in a control group based on the collection site, and try to reduce those effects.
Okay.
I see one that just came up and asked about global approaches on how will false discovery rate be considered? I can just approach that right now. What we do provide is we provide Q values in addition to the P values. I myself am not a statistician, but I'd be happy to, if you reach out to your BDE in your area, or even actually I'll just throw this up right now to the hello@metabolon.com. We can provide a kind of a more in-depth answer of how we develop those Q values and how that can impact your, how you can incorporate that into kind of false discovery rate.
Thank you. How do we see lipids in our global platform?
We see quite a few lipids in our global platform. So Metabolon, in addition to having a global platform, we also have a complex lipid platform, but I'll start out with the global platform first. The global platform will detect monoacylglycerols, phospholipids, sphingolipids, diacylglycerols, as well as lipid metabolites. So that would be, things, metabolites associated with beta oxidation. Those are acylcarnitines, bile acids, cholesterol metabolites. And so we do get a very good coverage of that to at least give you an idea of if complex lipids are being impacted. Now, if you have a more specific question and are very interested in triglycerides or cholesterol esters, we do have a complex lipid panel, and this panel covers complex lipids on the species level. So you're going to get your phospholipids, your sphingolipids, your triglycerides, cholesterol esters, diacylglycerols, and monoacylglycerols in one test. It's a single point quantitation, and the results are reported on a species basis as opposed to potential isobars.
How about short chain fatty acids?
We can detect those. Those are in our libraries, but they're not detected with 100% degree of on all studies. But we do have a targeted platform, which I referred to earlier in the talk. So our targeted assay team has developed a short chain fatty acid panel that is often used in conjunction with the global panel to kind of get an even bigger biological readout.
Thank you. And are we able to process COVID-19 studies or research?
Yes, we are still completely open for business, and we can process samples from patients that are COVID-19 positive. Again, this is one of those things we're very, very open to working with people to kind of understand this horrible disease. And so we are open to having these conversations and running these samples. We just like to know up front so that we can make sure our folks in the lab are well taken care of and are being as safe as possible.
I would just like to add, we have a COVID specific talk, and we do have a couple studies and I believe some announcements on those collaborations relating directly to COVID.
Thank you. Well, we do have a few other questions. I think we'll just take those offline, as I know that we're sort of running up out of time. What types of samples?
Oh, I can quickly go through some of these questions that I see. Okay. Yeah. So, all right, COVID. Right now, we've been doing plasma, but we can do BALF. We can any biofluid. Urine is all acceptable for COVID-19 clinical samples. So far, I think we've run. I think plasma has been the primary sample type that we've processed so far. One of the questions here is, the next two questions are any options to have kits where patients can collect their blood sample at home, like with the fingerprint prick, etc., and use it for global metabolomic assays? Yes, we actually have developed dried blood spot, a technology to use our global platform with dried blood spots. And so if that's something of interest, I definitely encourage you to reach out to either hello@Metabolon, Michael, or myself, and we can make sure to put you in touch with the right people to get that study off the ground. And then the last question I see here is, how many clinical correlative studies has Metabolon done with regard to respiratory fluids? And I don't know the exact number, but I know we actually have a case study that's going to be kind of recorded and put on our website soon, where they used nasopharyngeal samples, washes in infants, and looked at a metabolite signature that could predict the need for ventilators versus those infants with respiratory disease that did not need ventilators. So that's something that has been done. If you need more specifics, again, you can reach out offline and we can address that. And then I see one question over here in the chat asking about, do you work with Drosophila? And yes, we do. And that would be, I don't know the exact processes, but we do have standard operating procedures around kind of working with the Drosophila to process them through our system. So that's something definitely we can be done. We'd probably be looking at around 50 milligrams of flies. But again, I encourage you to have a discussion with the study director to really nail down exactly what your needs are. But we do have quite a bit of experience with Drosophila and insects in general. That is all I see, Carolyn.
Thank you, Carrie and Michael. Well, everyone, if you have specific questions for Carrie and Michael, their contact information is there. If you have about the webinar, if you have more general questions or questions about projects that you have worked with us in the past, feel free to reach out to us at hello@metabolon.com, and we will direct your questions to the best person who can answer those questions for you. Again, thank you very much for attending.
Thank you, Carolyn.
Thank you kindly.
Thank you.